So work on heat shock protein 60 with Ulrich was an exciting development for us. It started with study of a mutant of yeast in which we looked at proteins being imported into mitochondria. Most of the proteins that make up our energy producing organelles, mitochondria, are encoded in the nucleus, translated in the cytosol, and then imported as unfolded polypeptide chains, sort of like strands of spaghetti, across the mitochondrial membranes, and then fold on the inside aspect of the mitochondria to their native active forms. We asked the question in my lab with the student Ming Cheng whether it was possible that proteins might need assistance to refold inside mitochondria. To that time, people thought it was a spontaneous event. This was based on experiments carried out in the late 50s and early 60s by Christian Anfinsen and his co-workers, in which they unfolded a polypeptide ribonuclease in a test tube and asked whether it could find its way back to the native active form spontaneously. And it did. He received a Nobel Prize for that spectacular experiment. What we asked was whether there was such a thing as a folding machine inside mitochondria that refolded the newly imported proteins. When we tested our favorite reporter protein and one other protein, we were shocked to see that they failed to reach the native state in one of our mutants of yeast. We didn't believe it and we didn't think anybody else would, but a phone call from Walter Neupert and his associate, or Ulrich and his associate Walter Neupert, opened the possibility that they could help us biochemically to assess whether indeed we were looking at fully imported proteins that were failing to fold. And indeed, we sent our mutant to Ulrich, and he confirmed that that was the case. He and his co-workers then proceeded to reconstitute folding by HSP60 inside mitochondria, and using a bacterial system, George Lorimer and his co-workers showed that bacterial chaperonin, another ring assembly, a double ring assembly, and its co-chaperonin GRO-ES could be used to refold proteins in a test tube. This sort of summarizes the action of molecular chaperones in the cell. So the implications are that there are folding assistants inside cells that bind misfolded and non-native proteins, and in so doing, can prevent them from being toxic to the cell or preventing them from lacking any normal function. Overexpression of molecular chaperones that could recognize these proteins might be one way to resolve neurodegenerative conditions. But in another instance, binding by other kinds of components may also be able to be helpful. The most dramatic being the recent use of lecanemab to bind A-beta peptides in patients with Alzheimer's, which has shown some improvement for those patients and clearing of amyloid plaques from their central nervous system. So other kinds of approaches to clear toxic proteins may become accessible with time now.